Important fields in microbiology is agricultural microbiology. There are millions of microorganisms which are present in soil. For example, bacteria, actinomycetes, fungi, yeast, protozoa, algae, and cyanobacteria or photosynthetic bacteria. All these microorganisms are very important in agriculture. These microorganisms are of two types. Few microorganisms are beneficial in agriculture. They are responsible for increasing the fertility of soil, whereas few microorganisms are pathogenic to plants. That means they are responsible for the production of diseases in plants and crops. So, this study of phytopathogenic microorganisms, pathogenic microorganisms to plants, it is studied under plant pathology, which is a separate branch in agricultural microbiology. So, in general, we study the microorganisms which are associated with beneficial effects in agriculture and through crops. So, these microorganisms are responsible for increasing the fertility of soil. What is soil fertility? It is the ability of soil to support the plant growth. That means if the soil is highly nutritious, means if it contains all the essential minerals, elements responsible for the plant growth promotion, then we call that soil as fertile soil. And this addition of nutrients is possible by microorganisms. That means microorganisms are responsible for the addition of these nutritious elements, minerals to the soil, and then these elements are absorbed by plants. So, supply of essential minerals and growth factors, it is very important to protect the plants from pathogenic microorganisms by inhibiting their growth due to production of. So there is a category of microorganisms that supply nitrogen, phosphorus, and other essential elements. So this is how they promote the plant growth. There is another category of microorganisms which is responsible for the killing of different pathogens that cause diseases in plants. These microbes are called as biocontrol agents. These biocontrol agent produces antimicrobial substances. Nitrogen fixation. Nitrogen is one of the essential elements for the plant growth. Nitrogen fixation is carried out by microorganisms such as Azotobacter, Rhizobium, Frankia, Anabena. All these are able to fix nitrogen present in atmosphere and it is supplied to plants. We'll study that in detail in next few slides. Supply of phosphorus. Phosphorus is again second essential element required for plant growth. It is supplied by microorganisms through a process of phosphate solubilization that we'll be studying later on. So all these microorganisms that promote the plant growth are beneficial, and these beneficial microorganisms are supplied to plants in the form of biofertilizer. Biofertilizer is a preparation containing living cells of efficient microbes that have ability of nitrogen fixation, phosphate solubilization, and cellulose degradation. Microorganisms are useful in the control of insects that eat plants. Preparation containing such type of microbes is called as biopesticide. So biofertilizers and biopesticides are very important in agricultural microbiology. Genetic engineering or recombinant DNA technology that we have studied earlier is very important in agriculture, and that has brought new advancements or improvements in agriculture. Rhizobium meliloti. This is a bacterium that has been modified by genetic engineering for enhanced nitrogen fixation. Bt cotton is a variety of plants that have toxin producing gene from Bacillus thuringiensis. It is responsible for production of toxin, and this toxin kills the insects that eats plants. In this fashion, these plants have been made resistant to insect pests. Another example of application of genetic engineering in agricultural microbiology is a herbicide resistant crop variety has been developed that allows use of herbicide on weeds without damaging the crops. Several animal husbandry products have been produced by using genetic engineering technology such as porcine growth hormone, bovine growth hormone that improves the weight and milk production in animals. And these are produced by recombinant E. coli, that is genetically engineered E. coli. Plant pathology. It is another branch of agriculture in which it deals with the cause, resulting losses, and control of plant diseases. There are several objectives of plant pathology: to study causes of disease or disorders in plants, to study the mechanisms of disease development, to study the interaction between plant and pathogen, and to develop system of management or to avoid the loss of agricultural crops. That is all about the agricultural microbiology. A part of agricultural microbiology will be studying in detail, and that is microbes as biofertilizers and biocontrol agents. That is, we'll be studying the role of nitrogen fixers and phosphate solubilizers in the plant growth promotion, and the role of Bacillus thuringiensis as a biocontrol agent. So we have seen that microorganisms are associated with the roots of plants which are present in soil. So the area around the roots of plants is called as rhizosphere. And in this rhizosphere, we'll find the presence of beneficial microorganisms. That means these are the microorganisms that are beneficial for the plant growth. The biofertilizers or biocontrol agents are used for the improvements in agriculture. These microbes that supports the plant growth, these microbes which are responsible for increasing the soil fertility are called as PGPR. That means plant growth promoting rhizobacteria. There are several examples that you need to remember: Pseudomonas azospirillum, Burkholderia, Bacillus, Enterobacter, Rhizobium, Irvinia, Serratia, Alkaligenes, Arthrobacter, Acinetobacter, Flavobacterium, and many more. All these are the names of genera. What is a biofertilizer? That we have already seen. It is a preparation containing live cells of efficient strains of microbes like nitrogen fixing microbes, phosphate solubilizing microbes, and cellulolytic microorganisms. So these soil microorganisms are responsible for increasing the fertility of soil, that is its ability to support the plant growth. So these microorganisms are responsible for the supply of essential minerals and growth factors, and these essential minerals and growth factors are supplied to plant that improves its plant growth. So the first essential element is nitrogen. Nitrogen is freely available in air, but plants cannot utilize that atmospheric nitrogen. So aerial nitrogen, that is N2, cannot be utilized by plants. So therefore this nitrogen has to be converted into cellular form. That means this gaseous nitrogen should be converted to cellular nitrogen. Cellular nitrogen means the form of nitrogen which can be utilized by plants. Gaseous nitrogen cannot be utilized by plants. Cellular nitrogen is utilized by plants, that is present in the form of ammonia. So conversion of gaseous nitrogen into cellular nitrogen, that is ammonia, it is called as nitrogen fixation. And this nitrogen fixation is carried out by microorganisms through the action of enzyme. That enzyme is called as nitrogenase enzyme. Then the product of nitrogenase enzyme, that is ammonia, it is then absorbed by plants. This is how the nitrogen fixation process occurs. There are several examples of nitrogen fixers. These are divided into two categories: free living nitrogen fixers. Free living means in absence of any association, these microorganisms can convert gaseous nitrogen into ammonia. These examples are bacteria such as Azotobacter, Achromobacter, Bacillus, Chlorobium, Chromatium, Clostridium, Pseudomonas, and many more. Another category of nitrogen fixers is symbiotic nitrogen fixer. Symbiosis means association. So these are the nitrogen fixers or microorganisms that can convert gaseous nitrogen into ammonia only when they are associated with plants. That is why they are called as symbiotic nitrogen fixers. Example is Rhizobium. So Rhizobium, it fixes nitrogen only when it is associated, or only when it is present in root nodules of leguminous plant. If you see the example of pea or bean or fenugreek, or similar leguminous plants. So if you see the roots of these leguminous plants very carefully, you will find that there are presence of root nodules, म्हणजे गाठी असतात. And if you see carefully, the color of these root nodules, it is pink. So that pink color is due to presence of leg hemoglobin, which is a sign of presence of rhizobium. Another essential element after nitrogen which is required for the plant growth promotion is phosphorus. Generally, soils are deficient in phosphorus, or whenever this phosphorus is present in soil, it is present in a complex form. That means it is present embedded, immersed inside the soil particles or inside the rocks. Therefore, this phosphorus is generally supplied as a chemical fertilizer in the fields. Phosphorus is present in rocks and soil in the complex form, and it cannot be made available or it cannot be absorbed by plants. So it is unavailable for the plants. So there are microorganisms which makes this phosphorus available to the plants. That means these microorganisms are responsible for releasing phosphorus from inorganic and organic sources, that means from soil and rocks, through the process of solubilization. Phosphate solubilization, it is the process in which complex phosphorus source is converted into simple by means of microorganisms. These microorganisms are called as PSB, phosphate solubilizing bacteria. The mechanism by which they solubilize or they make this phosphorus available is through the production of organic acids. So these organic acids which are released by this phosphate solubilizing bacteria, they convert this insoluble form of phosphorus present in rocks and soil into soluble form, which is then utilized by plants. Examples of phosphate solubilizers are for Pseudomonas, Bacillus, Rhizobium, Burkholderia, Achromobacter, Agrobacterium, Micrococcus, Aerobacter, Flavobacterium, Erwinia, etc. So, why these nitrogen fixations are very important? So, why these nitrogen and phosphorus through microbes is important? Because there is another way of supplying nitrogen and phosphorus, that is through the supply of chemical fertilizers. But there are disadvantages of chemical fertilizers, that is negative impact on environment, and these fertilizers are very costly. Therefore, the eco-friendly and cheap method is the use of PGPR for the maintenance of good agricultural practices. Nowadays, we call it as organic farming. Another area which is important in agriculture is biological control, or it is also called as biocontrol. As the name indicates, control. So this is associated with the control of diseases in plants or in agriculture. So what are the agents which are responsible for production of diseases in agriculture, or what are the agents which are responsible for the destruction of crops? There are two. First one is pathogenic microorganisms. They are responsible for the destruction of crops. And second one is larvae or insects. These insects are responsible for the eating of plant material and they destroy the crops. So there are two approaches in biological control that we are going to study. So in general, biocontrol is the use of organisms for the management of plant diseases. Plant diseases can be controlled by other methods also, that is by the spread of fungicides, pesticides, bactericides. That means by the spraying of chemical agents, but these pesticides or bactericides are harmful for human health. Therefore, biological control is a very eco-friendly approach in which microorganisms are used for the killing of insects, or microorganisms are used for the killing of other pathogenic microorganisms. So the agents that are organisms that are used in biocontrol are called as biocontrol agents. So these biocontrol agents are responsible for the killing of insects, and they are responsible for the killing of pathogens. These microorganisms or biocontrol agents are safe for in terms of environmental point of view. That means they are not pathogenic to human beings. In entomology, it is the study of insects. So in entomology, the biocontrol agents are live predatory insects. So these are live predatory insects, entomopathogenic nematodes, or entomopathogenic microbes. So in these three categories, live predatory insects, it is a part of zoology. Entomopathogenic nematodes, it is a part of zoology. Entomopathogenic microbes, it is a part of microbiology. Entomopathogenic microbes means these microbes are responsible for the killing of different insect pests. So these microorganisms control the insects that eat the plant material. So these preparations containing entomopathogenic microorganisms, it is called as biopesticide. So the definition is preparation containing insect killing microorganisms. It is called as biopesticide. So which microorganism is used in the preparation of biopesticide? It is Bacillus thuringiensis. It is a widely used bacterium in the preparation of biopesticide. Why this organism is used? Because this bacterium, it produces a toxin, and this toxin, it kills the insects only. There is no effect of this toxin on human health. Considering this, advanced techniques such as genetic engineering technique has leads to the advancement so that a resistant plant resistant variety, insect resistant variety has been developed. So what is that genetic engineering technology for the making of insect resistant plant variety? As we have studied earlier, that what happens in genetic engineering, that this Bacillus thuringiensis, it is responsible for the production of toxin, which is a protein. If it is a protein, then this organism must have a gene inside its chromosomal DNA. So the gene from this Bacillus thuringiensis is isolated, and then this gene is transferred into the plant variety. For say for example here, a cotton variety. So a gene from bacterium is cloned into the plant cell, that is cotton plant cell. So when this cotton plant grows, when the cells grows, these cells will start production of this toxin. So whenever that insect, it eats this plant, it will try to eat the plant variety, then what will happen? This toxin which is present in this plant material, it will be ingested by the insects, and insect will be killed due to the action of this toxin. So that is an excellent example of genetic application of genetic engineering technique, that is the development of bt cotton variety. So this bt cotton variety has a gene from Bacillus thuringiensis. That gene is responsible for production of toxin. So this bt cotton is a variety of plant that has a toxin producing gene from Bacillus thuringiensis. So Bacillus thuringiensis is widely used bacterium in the preparation of biopesticides because it produces a toxin, as we have discussed, and its application in genetic engineering for the development of resistant plant varieties we have also discussed. Then another field in agricultural microbiology is plant pathology. So, in plant pathology, the term biocontrol applies to the use of antagonistic microorganisms to suppress the growth of diseased plants. So, what happens in antagonism? Antagonism means one organism is producing or one organism is killing the other organism due to the production of toxic substances or antibiotics. So here antagonistic microorganism is killing the disease causing microorganism, that is biocontrol. Example of biocontrol agents in this context are trichoderma. Trichoderma is a filamentous fungus, and worldwide trichoderma is used as a biocontrol agent because it suppresses the diseases of plants. It kills the other plant pathogenic fungi. This is also a fungus, but it kills other plant pathogenic fungi. So, which is an excellent example of biocontrol agent. Other examples of the species of trichoderma are Trichoderma hertzianum, Trichoderma viride, and Trichoderma aspergillum. These are the widely used species of trichoderma which are used as biocontrol agents for the control of fungal plant pathogens. Along with trichoderma, there are certain bacteria that are used as biocontrol agents. So they are killing the other pathogenic bacteria and pathogenic fungi, plant pathogenic fungi and plant pathogenic bacteria. These biocontrol agents are Bacillus, Pseudomonas, Enterobacter, Lysobacter, lactic acid bacteria. So all these are useful in the making of biocontrol agent preparations.